Welcome to this presentation on Nanolive live cell imaging. What you see here is an example of a dataset you can acquire with Nanolive imaging. You can see cells in the environment roaming around, you can see the adherence between cells on the dish, and you can see the internal structures. All of this is completely label-free. So the first question I have for you is, what are the techniques you currently use to image your living cells? They are often techniques either in phase imaging or fluorescence imaging. These are both great depending on what you're trying to look for, however, they both have their own limitations. In phase imaging, you are limited to a lower resolution and 2D information which you cannot quantify easily. With fluorescence imaging, you can get this quantification and high resolution, however, you can encounter problems of phototoxicity. So that's where Nanolive comes in. The idea is to bridge the gap between these two worlds and provide a label-free imaging solution which can allow you to image your cells for a long term, at a high resolution, and without events of phototoxicity. Let's have a look a bit deeper at what kind of structures we can see with Nanolive imaging label-free. Here's an example of human primary keratinocytes. Here you can see the nucleus, the nuclear envelope, and the DNA inside the nucleus. You can see as well mitochondria and the mitochondrial dynamics, fission and fusion. And along with that, in metabolic studies, you can follow the lipid droplets, which are the shiny white dots that you can see here. The more diffused dots are lysosomes, involved in all kinds of events, and you can have here the vacuoles as well in these darker spots that you see inside the cell. This is an example of the membrane, adhesions, protrusions, and just the general dynamics that you can achieve with Nanolive imaging. As a reminder, this is all label-free and it's 3D information. What you see here is a maximum intensity projection in Z just to visualize all of the structures in a single plane. But what you achieve is always a 30 micrometer depth of imaging. The resolution is 200 nanometers and is completely non-invasive. To highlight this 3D capability, we have used here a different gradient from blue to pink in the Z-axis. Here you can see then the adherent cells, the cancer cells at the bottom of the dish, which have a blue surface, and as you go higher and have the T cells floating above these cells, they are in pink. This is just to visualize the three-dimensional situation, but what we will always use in our presentation is refractive index values, because that is what we obtain with our microscope. Here you have an example of T cells killing a target cell, and you see them in a grayscale value where you see in higher density, so the T cells, which are nice and round, in white, and you see the target cell with different gradients of refractive index. And you can see here that as the cell evolves and goes through the process of cell death, the gradient changes and the density changes as the membrane starts blebbing and the internal structures decompose. So why is the refractive index important, and why is that what we chose to pick up with our microscope? Here you have an example, just to simplify the concept, of a mountain that you want to take a picture of on a summer day. So say you want to take this picture, you will have the light that travels to the objective, and it goes through air, which is a very homogeneous distribution of refractive index. So the resulting image that you will get is a picture of the mountain as you see it. Now, if we take the same situation, but instead of having this homogeneous distribution of refractive index, we add something in the middle to make it non-homogeneous. So for example, a glass of wine. So here, as the light will travel through the air, the glass, and the liquid, and then reach the objective, the resulting image will be distorted because there has been a shift in the phase of light. And this is the property of light that we use with our microscope to obtain our images. It's the fact that the light remembers the contributions of the refractive index along the light path. Here, if we take it now to a more technical point of view, you have a light path, which is a reference field with a very clear pattern of maxima and minima. And then you'll have a certain distribution of refractive index, taking again this example of the glass of wine. As the light will travel through this distribution of refractive index, there will be a change of the phase of light, and you will get a sample field, which will have a different pattern, and it won't be as regular as the reference field. The idea is that if you can get the information of the reference field and the sample field, you can, through computer processing, obtain information on the refractive index through reconstruction. So then at the end, you have an image that represents the refractive index values of the distribution that the light went through. And you can get quantitative values from this, as each pixel has a value associated to it. And this is exactly what we do with Nanolive imaging, except that instead of having a glass of wine, we actually have cells in the middle. And as each cell has has different compartments and different organelles, which all have different refractive indexes, this is how we can then obtain images of the cells in high resolution. Let's have a look at the microscope itself. This is what the microscope looks like. It has an objective at the bottom, and the sample sits above the objective. The light comes from above, and it is actually split into two so that you have the reference beam and the sample beam, exactly how we had in the example before. If we look inside the microscope, that's what we see. You have on one hand the light source in the top corner, split into two, sample beam, reference beam. As explained earlier, the reference beam will not be distorted, however the sample beam will. And as the two meet together, you can then create an image of the interference pattern and the holograms that come from that. So that is basically the principle of holography, is to determine the differences in light phase shift. Now, where Nanolive is unique is that you might have noticed that instead of having a light coming from above, it comes at an angle with this rotational arm that you can see. Sending the light at a 45-degree angle not only helps us achieve a higher numerical aperture and therefore a higher resolution, but by turning around as well around the whole sample, you can collect more information, and that's the principle of tomography, where you assemble information from multiple angles. So therefore, we're achieving higher resolution and 3D information. And this is the kind of image we get at the end, a gradient of the refractive index from dark to light. And as the cell evolves, like here, the cell going through mitosis, you can see that the chromosomes get more denser and then they get whiter. And again, as it separates into two daughter cells, they go back to the original gradients. So essentially, this is really one of the only systems that can allow you to see mitochondria completely label-free. Here you have an example of a zoom-in of certain mitochondrial dynamics, fusion, fission, and it's completely without any markers. So you know that you're really seeing the cells in the healthiest state possible. So how do we know that these structures are indeed mitochondria and also all the other structures I spoke about earlier? Well, it's because we have a correlative epifluorescence module that is connected to the microscope, so you can overlay of your fluorescence channels with the refractive index values, and this can confirm the structures that you're looking at and really show you where your structures are within the cell. This can also help to tag certain structures that we cannot see with the refractive index alone, and you can then see where they are within the cell and follow their evolution. The laser power that we use is extremely low. It's 20 milliwatts per centimeter squared, which is 1/5 of that of a light sheet, which is known to be one of the more gentle techniques. So that is why we can image stem cells and sensitive cells for a very long time. So up to 48 hours imaging has been achieved, and they do not escape the light, they do not get damaged by the light due to this low laser power. And that is how we can achieve these kind of images, for example, of primary cells. And also you can then see stem cell differentiation over long periods of time. So essentially, if you can see what's happening on your living cells, you can see the effect of drugs directly on these living cells. Here we have an example of a drug candidate which is rescuing a cytotoxic effect induced by BSO. You can see here on the left-hand side where the cell is dying, the mitochondria are fragmented, it's losing its adhesion and really getting more compact, which are all signs of cell death. And on the right-hand side with the drug candidate, the cell is healthy, it is happy. You can really compare these two conditions and have a direct readout of your drug effect. This can be done in a multiple well format. You can enhance the amount of conditions that you're looking at. You can do with and without fluorescence. There's many kinds of options that you have available. And this is thanks to the automated stage that we have associated to the microscope. This automated stage is not only to multiply conditions, but it is also to enhance a field of view. As the standard field of view is 90 microns by 90 microns, this could be limiting in certain assays, so there's a possibility to do a grid scan where you can then stitch up the images together, and here's an example of a 3x3 grid scan. You can see that the cells are very fluid between the different sections of the grid, and there's no strange jumps. You can also, if you're working in dishes, go up to much larger fields of view, and this is an example of a 10x10 grid scan. Just to keep in mind, the image resolution is always maintained. So what do we do with these images once they are acquired? Well, there are some analytical and quantitative tools that are provided to be able to make sense of this data. Here we have an example of our segmentation algorithm, which works either on single cells, as you see here on the single pre-adipocyte, or as you increase complexity of the cells in different confluence, you can still achieve a very clear map of these cells. Once this mask is achieved, you can then get out single cell metrics, for example, both quantitative and qualitative. Here we have an example of a control cell in green and a treated cell in orange. And you can see how there's a clear difference, a clear cell death phenotype, but it can also be quantified by checking the cell perimeter changes over time, the cell extent, and the cell area. This can also work with multiple cells. Here we have a more complex population. We have in green pre-adipocytes with a certain inhibitor, and we have in orange glioblastoma cells with that same inhibitor. And the idea is to see how the effect is different. You see on one hand that the cell death itself is different, as in green it was more apoptosis. However, in orange, we are seeing events of necroptosis. So this is the qualitative information that you can get, and beneath you can see the graphs of the evolution over time of the cell population in terms of the cell count, the cell area, and etc. And this really provides you quantitative information to support your data. The segmentation can also be done inside the cell with the different organelles. So here's an example of the lipid droplets, which have been segmented with the algorithm based on the refractive index. And you can see that it works very nicely. You can get some tracking then of each individual lipid droplet. This can also be extended to multiple cells. As you can see here, each lipid droplet in each single cell has been segmented, and this can give you a vast array of data. So that's just an overview of the different applications for which Nanolive can provide a clear added value. You can see that it's for multiple fields of application, from neuroscience to metabolism to immuno-oncology. And here are just a few examples of some beautiful cells that we can image with Nanolive imaging. So to summarize, this is really a very easy way to get quantifiable data without any phototoxicity or photobleaching, and with a very fast acquisition speed, picking up on fine dynamics and interactions between cells. And it's with the very intuitive design and software, easy to use, user-friendly, and a brand new way to image your cells.